Hello everyone, welcome to the next module. In this module, we will be discussing the topic which is dendritic cells, and the next one is NK cells. So we have already covered some of the cells before, and in this particular section we'll be covering dendritic cells and NK cells. Let me draw the dendritic cell first. In the dendritic cells, what you see, these structures, these long projections, they are called dendrites. And these dendrites are really important for efficient antigen capture. Okay, so let me label them. These are the dendrites, and they are really important for efficient antigen capture because they increase the surface area. So high surface area is usually provided because you have so many dendrites. And then in the center you have nucleus, and nucleus is irregularly shaped nucleus. So that's the feature of these cells. Next, what you have is let's talk about the granules, and let's talk about the structures that are there inside the cells. These are for antigen presentation. They are also known as endosomes. So endosomes are required for processing the antigen. So these are some of the important structures that are there in the dendritic cells. Now let's talk about the receptors. Now what you'll have is, the first one is MHC class 1 receptor, which is really important for self-recognition. Then you have MHC class 2, MHC C2. I'm writing C2 over here, and these receptors are really important when it comes to antigen processing, okay, and displaying the antigen to the T cells. Next, you have CD8, CD80 cluster of differentiation, 80 receptor. This is also one of the important receptors. So let me write it down. And another one is also there. Is another CD receptor is there. So this is CD8080, and this one is CD. I don't have space over here, 86, okay? So CD86 molecule. I'll explain these receptors in detail, but anyways, you need to know. Another receptor that is there is toll-like receptors. Toll-like receptors are receptors. They are really important when it comes to recognition of pathogenic molecules. Now let's talk about these receptors in detail. One is MHC class 1 and MHC class 2. Let's talk about both of them. So let me write MHC class 2 also over here. These two receptors are important for antigen presentation. So antigen presentation specifically to T cells. Antigen presentation. If I diagrammatically try to show you, so let's see if this is the dendritic cell, and that's a nucleus. It will have the receptor, which is MHC receptor, with additional receptors are also there. It will interact with another cell, which is the T cell in this case, and there will be another receptor from T cell side. This is MHC, and there will be some co-receptors along with the MHC, and in the T cell you will have that particular receptor from T cell. They will interact through that antigen which you're seeing in the center, and then sudden activation of the cell will happen, right? And then after that, you have another receptor, which is CD8086 receptors, and they are the co-stimulatory molecules that are required. So let me write it down: co-stimulatory molecules, and they're required for full activation of the T cells. So full T cell activation is really important. Next one we have is a very important receptor that is there on the dendritic cell, toll-like receptor, TLR. And that's a full name, which is toll-like receptors. And they are really important for LPS recognition, DNA recognition, RNA recognition, and they're also required for any other protein molecules or antigens that are there in the pathogens. So they are kind of a gate checkers. Okay. Now, if we talk about the functions of dendritic cells, as we are discussing dendritic cell first, the main important point is they are professional antigen presenting cells. Okay, they are the professional APCs. So APCs, in short, antigen presenting cells, and the function that they perform is phagocytosis. And let me create the illustration to show you the phagocytosis, where bacteria, let's say this is the dendritic cell, and these are some of the dendrites, and what it does, it actually takes the bacteria inside. So as you can see over here, now bacteria is inside a particular structure, and then it gets destroyed inside the cell, and then the part of that particular bacteria either processed or expelled out. And this is the process, phagocytosis. Although this process is pretty much elaborated and complex, but at least now you know how phagocytosis works, right? So this is really important process of dendritic cell. Now, if we talk about MHC class 1, they usually interact with CD8 positive receptors that are present on the cells, T cells. In MHC class 2, they interact with CD4 positive T cells, and they are helper T cells. Okay, the top T cells are cytotoxic T cells. We will know about these things also in more detail in our next course, which is on adaptive immunity. Now, let's talk about migration. These dendritic cells, they also migrate. Okay, migrate to different tissues. They migrate to lymph nodes. And so let me write it down, migration to lymph nodes, and they require a particular receptor for that particular migration process. So that receptor is really important to know, CCR7 receptor, CCR7. The full form is chemokine receptor type 7. So there are so many different receptors are there in the cells. One of the important receptor that is required for lymph node migration is chemokine receptor. And it's also known as C-C because two cysteines are involved in this particular interaction. So C-C. Next, what it does, it guides the migration of these cells to lymphoid tissue. So there is always that interaction that is going on between the cells, and the cysteine plays very, very important role. To know that two cysteines, they are going to interact, and then that is how the receptor is going to interact with its like in molecule, and then migration of the cell is going to happen to the lymph node. As you can see over here, we have covered not only just overview of the dendritic cell, but also the functions in more detail. We talk about CD80, CD86 costimulatory molecules. We talk about LPS receptor, which is toll-like receptor, and we talked about CD8, CD4 receptors, and then another important receptor is CCR7 receptor. So I want you to actually remember these receptors and their function so that you can understand what is the structure as well as the function of dendritic cell, right? Now let's move on to the understanding of the NK cell. Moving to the next part of this particular lecture is natural killing cells. They're also known as NK cells. So I'll start with the creating the structure of this particular cell. This is very large granular lymphocyte. So it contains granules, right? It's a large lymphocyte. So that is what I've written over here. Next is when we talk about the nucleus. Nucleus is round in shape. And it's irregular in dendritic cells, right? But in NK cells, it's round, and it has these granules that I'm designing over here. These are also known as cytotoxic granules. So these granules are really important because this cell requires the ability to kill the other cells that that are not good for our body, right? So it contains cytotoxic granules. Next, now let's talk about the receptors, and we'll also talk about the receptor in detail. Let's understand that what kind of receptor we have. CD56 is one receptor that we have. Next, I'm going to draw very close to this is another CD receptor, which is CD16. It's also known as Fc gamma R3. Okay, Fc gamma R3 receptor, very important receptor for antibody interactions, and I'll explain it when I explain all these receptors after some time. So next is what you have is you also have another receptor which is known as KIR receptor. So let me draw right over here, KIRS receptor. Next receptor we have is another interesting receptor, which is NKG2D receptor. Okay, very important receptor. Now let's understand what are these receptors. So we'll start with these surface receptors or markers. Let me write it down. The first one, which is CD56. CD56 molecule is also known as cluster of differentiation 56 is required for adhesion to by the NK cells because it need to attach. Next one is CD16, and as I told you, this is Fc gamma R3. And Fc is coming from the Fc fragment of antibody. For that, I need to create the structure of antibody. So let's suppose this is the antibody, and I hope you remember it has Fc region. This is the Fc region, and this particular receptor basically binds to this Fc region. So that's why it's known as Fc gamma R3 R3, and R3 is basically receptor, and 3 is the type of that particular receptor. Next is KIR, which is killer cell immunoglobulin-like receptor. So they are killer cell immunoglobulin IgG-like receptors. And they are important because inhibitory receptors, they play a very, very important role in recognizing MHC class 1 proteins, right? So recognition of MHC class 1 is completed by this. Next is fourth one, NKG2D receptor. NKG2D receptor is also known as natural killer group 2 member D receptor. So based on their classification, they are named like this. And it's really important because it helps in activating. It's a receptor that detects the stress signal on abnormal cells, stress signals. So abnormal cells are basically detected by using this receptor. Now, if we talk about the function is very, very important of natural killer cells. As you can see, as you can understand by the name, it can kill the cells which are virus infected cells. So viral immunity is really important. Next is you have tumor cells. Tumor cells are the cells that have that ability to grow uncontrollably. So cancer immunity is very, very important when it comes to natural killer cells. So NK cells are really important with virus infection, with tumor cells, or in cancer. Next is it also detects the missing MHC class 1 receptors. If this particular receptor is missing in the cell, it also detects that and categorize those into abnormal cells where you don't have MHC class 1, and then goes and kill the cell. So this is how it takes care of the cell, which is not the part of our body if these cells are defective. Now, if we talk about the mechanism of the killing by NK cell, it's really interesting that how this cell kills the other molecules, other pathogens like other cells also. So what it produces the protein called perforin. Perforin is the protein that can actually create pores. Okay, it can create pores inside the target cell. So suppose if this is the cell, it can actually create pore on the surface of these cells, and because of that, the cell is eventually going to lyse. Next is gran enzymes. Now, when pores are created, next point is gran enzymes, they can enter inside the cell through these pores, and they trigger apoptosis, which is essentially the killing of the cell, right? That will lead to the killing of the cell. And what happens is the entire cell basically collapses. Entire cell is going to collapse into smaller vesicles, and it's not messy. So it's very controlled process, but ultimately it leads to the death of the cell. So that is something really important function of the natural killer cell. That is why they are also known as natural killer cells, right? These are some of the basic functions. Now let's come on to some of the advanced functions of natural killer cells. We'll start with the FAS and FAS-L pathway. FAS and FAS-L pathway, and what that pathway is, let me explain to you. But let me tell you that what is the FAS and FAS-L. We'll start with FAS. FAS is nothing but it's a CD, cluster of differentiation molecules, CD95. So the FAS protein is also known as CD95, and this is a death receptor. This is a death receptor on the target cell. So that receptor is recognized by NK cells, and for NK cells, it also requires a ligand receptor, which is FAS ligand, FAS-L receptor, that is present on the NK cell, right? It's also known as CD95L. So this protein is present on NK cell, and this protein is basically utilized for interaction with the FAS protein. Now, if I diagrammatically try to show you, suppose this is the NK cell, and NK cell will have a receptor on the surface. Suppose this is the receptor, and this receptor is CD95L receptor. Understand, right? And then if let's say we have a target cell right over here, this is our target cell. The receptor target cell, the receptor that is present on the surface, which will interact with this CD95L, is the FAS receptor or CD95. So let me write down FAS also, FAS receptor. Now FAS will interact with the FAS-L, and because of that is going to recognize and then initiate the process of apoptosis, targeting the cell, trying to kill the cell. So this cell will be killed, and then usually the process is highly controlled. So this point is also important. Controlled. The process is controlled, so no inflammation will be there when the cell is getting killed. So no inflammation. That's the feature of apoptosis, right? Okay, so this is one of the advanced interaction that happens in case of NK cells. Now, second, mechanism is ADCC. ADCC is nothing but antibody-dependent cell cytotoxicity. Let me write it down. Cytotoxicity, cell cytotoxicity. Where what happens is, let me show you it in an illustrative way. So let me draw the NK cell. Let's draw the target cell first. So let's suppose this is the target cell. It will express that antigenic dot that you're seeing over here. That's the antigen, and antigen will be expressed, and antigen will basically bind with antibodies, right? Because that's how immune system works. Antibodies, they have the affinity. So this is your antibody, which is in cyan color, and now antibody will interact with the antigen, and then this process is known as coating. So basically what is happening, cell is getting coated. Now if you have enough antigens that are coated, what can happen next is basically there can be interaction between the FC receptor, which is present on the antibody, and you have FC receptor ligand, which is CD16, which is also known as FC gamma R3 receptor, and this receptor is present on NK cell. So NK cell will come and bind to that antibody. So this is our NK cell. It will come and bind to the antibody. Now what will happen? The NK cell will get activated, and through antibody, now it is targeting the cell and it will kill the cell. So the killing of the target cell will happen, and this cell eventually will be eliminated from the body. So this is known as antibody dependent cell cytotoxicity, means antibodies initiating the process of killing the target cell. Next one is cytokine associated NK cell function. So what is the role of cytokines? So NK cell also produces different cytokines. Let's talk about one of the important cytokine that is released by the NK cell. So NK cell, as I told you, it can produce different cytokines. So let me write it down. Cytokine will be produced, and the important cytokine that is produced is IFN gamma, interferon gamma. So interferon gamma is one of the important cytokines that is produced by NK cells. So these are the dots that I'm creating. These are the cytokines, and it will increase the macrophage activity. So NK cell can also enhance the activity of the macrophages. Next point is it can also cause enhancement in the immune response. Basically, it can boost the immune response of the body. So this is another role that NK cell plays. So you can imagine what kind of advanced role NK cell can, why they are so important for our immune system. So as you can see over here, we have covered the structure, the surface of dendritic cell, function of dendritic cell, as well as natural killer cells in detail. And then we also covered advanced topics like fast cell pathway, ADCC, and cytokine. So that gives you a lot of information on these particular cells, right? So that covers our next topic on our innate immunity course, because most of them they are the part of innate immunity. We are not focusing on antibody. Antibody is the part of the adaptive immune system, but NK cells, as you can see over here, they play very important role in our immune system. So we are covering all those topics right over here, and also trying to cover their advanced functions. All right. So before I conclude this, I'm requesting everyone, if you like the video, then please press the like button. It helps us to reach out to more and more students. And if you can share the video, if you can comment what is your, if you have any doubt and if you have any feedback, because your comments also help us to reach out to various students, right? So that helps us, that motivates us to create more courses that will help different students in their exams and all. In future, we'll be bringing more and more videos, but we need your support. Everyone needs support so to grow, basically. So if you can please support us by just liking the video, if you have understood the topic. If you have any comment that you want to make, please comment on the comment section. That will help us a lot. At the same time, I also want to introduce you about the platform that we have created. It's called Research Goal. If you type Research Goal, you should be able to visit the Research Goal website, where I host a lot of courses that are advanced research level courses, where I teach how to do advanced research, how to use those tools, how to do analysis, data analysis. A lot of different courses are there, and they are not that expensive. And those are practical based courses, right? They are highly advanced courses. Here you get a basic knowledge of the subject. There you get research skills. Okay, so I want you to please explore and see, because we also have free modules in those courses. They are absolutely free. You can actually see and evaluate and see. If you want to complete other modules, then you can join in. The amount that we are putting for these courses, barely, very minimum amount we are asking compared to other course platform. So I hope that will help you gaining the research skills that you want in the area of immunology, biotechnology, microbiology, and a lot of the computational biology courses are also there, which will help you. Okay, so I'll meet you in the next part of this particular course, and hopefully we'll be soon concluding the innate immunity, and then we can move on to another topic like that. Okay, so I'll try my best to actually give you amazing experience with these courses. And if you have any suggestions that you want to make, please feel free to contact us. Please feel free to comment and give your thoughts on the video that we create. Thank you so much, everyone. I'll meet you in the next one.